Okay, 3, 2. Hello, everyone. I would like to welcome you to this single-cell data analysis webinar by QIAGEN Digital Insights. Thank you so much for joining us today. My name is Andrew Olson. I'm the Associate Director for our Discovery Insights portfolio, and I'll be your host and moderator for today. Today's topic is about single-cell data in the context of biomarker discovery and target identification. Today, you'll learn about the importance of expert curation and how to explore cell resolution data to visualize within and across data sets in the context of your biomarker and target discovery questions and experiments. Our speaker today is Dr. Joe Pearson. Joe works as the Global Product Manager for QIAGEN OmicSoft. Before joining OmicSoft in 2015, Joe earned his PhD from the University of California, San Diego, and was a postdoc and research assistant professor at UNC Chapel Hill, studying cis regulatory codes during animal development. His presentation is called Rapid Cross-Project Single-Cell RNA-seq Exploration for Biomarker and Target Discovery. And with that, I will hand it over to Joe.
Thank you, Andrew. So before we begin, I'd like to mention that these QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. As a brief introduction to QIAGEN OmicSoft, our goal is to accelerate biomarker discovery by allowing bioinformaticians and biologists to quickly answer questions relevant to biomarker and target discovery. Through our LANS platforms, we provide the ability to discover curated disease-relevant omics studies and find experimentally validated biomarkers by exploring our collections of over 600,000 samples across 30 databases and from over 7,600 projects. By exploring these databases, you can reveal meaningful patterns for your biomarkers across diseases to do things like answer questions about where the expression patterns are between normal and diseased samples. You can explore across our data sets and use the platform to correlate multi-omic data and the extensive clinical metadata that have been curated. And you can use the platform to interactively explore the data at sample and now cellular resolution. The platform also allows you to export the full expression data, other omics data, and the metadata so you can perform your own downstream analyses with these curated data. In addition, through our platform, you can explore thousands of pre-computed statistical comparisons. These are comparisons built on the various projects that are found within the LAN databases that are curated to reveal differential gene expression between disease versus normal, treatment versus control, treatment responsiveness, tissue versus tissue, and more. These comparisons can be directly uploaded into QIAGEN IPA, and you can also compare your data sets in IPA to over 90,000 comparisons with IPA analysis match. The single-cell LANs that I'll be telling you about today are really just an extension of our existing collection of LANs and our LAN platforms. As with our other LANs, the goal is to enable two key workflows: to either explore across the projects to discover relevant data sets, or to explore by your biomarkers and find those projects that are most relevant to you. As an example, you might explore within the single-cell LANs and start at the project level and query our extensive curated metadata to filter down and regroup and organize the data to find the exact data sets and the relevant projects that you're looking for. For those projects, you can explore the deep curated metadata directly overlaying on dimension reduction plots and discover the specific data sets that are relevant and investigate the individual clusters that have been curated. As part of our curation process, we perform pre-computed comparisons between each cluster and all other clusters within the dimension reduction plots. And so you can explore these differential expression results to identify the top upregulated genes within each of those clusters. And then you can directly explore the expression of those at the cellular level on dimension reduction plots with gene expression overlays. The other key workflow that our interface supports is the gene-based exploration view. Perhaps you've identified some candidate biomarkers from your internal research studies, or by exploring disease land and oncoland and the extensive studies there. And you want to dig further into these results and understand the cellular context of these gene expression patterns. And so you can start with a gene and explore across all of the different projects that have been curated within single-cell lands and look for cross-project expression, organized across the different tissues, diseases, curated cell types, and more, organized on any of the curated metadata. You can also search for your gene of interest and look for upregulation and downregulation of your gene of interest within each curated cluster with the differential expression bubble plot. And then you can also finally inspect your gene expression patterns at the cellular level on any of the projects. And then take these data and download the full expression data sets along with the curated metadata and explore further. The key principle behind our single-cell lens framework is to enable cross-project exploration. And so this requires several key steps in our curation process to enable this. You're able to group, filter, and explore the gene expression details for individual studies and across studies because of our consistent terminologies that are used during our manual curation process. We use controlled vocabularies that are compatible with disease land and oncoland terms, and we curate dozens of different parameters available for different studies. During our curation process, we reprocess the data from raw files all the way through alignment, quantification, quality control steps, and differential expression analysis using consistent pipelines so you can compare more readily across the different studies. Our platform allows dynamic filtering and grouping on any of the metadata curated at the project level, the sample level, or the manually curated cell types identified for each cell cluster. So you can explore the individual data sets in detail, looking at this collection of similar data sets with similar cell types, or you can combine them together and visualize them side by side, integrated on any of the curated metadata. We have data sets available for both non-UMI and UMI data sets, and we have data sets curated on human and mouse. Through the interface, you can explore the curation at the project, the sample, and the cluster level. As an example of some of the project level metadata we curate, you can see here that we've curated the project IDs, the technical parameters such as the platform and the PubMed ID, the basic study type, as well as the overall theme of the study, such as the therapeutic area and the disease. So you can quickly find studies that you might be interested in based on the theme. You can also explore these studies at the sample level. What are the contents of the experiments that were performed? So over here, we can see on the right a number of samples from a type 2 diabetes experiment. And with each sample, we've curated the disease state, the tissue source, as well as extensive clinical parameters for each biological sample and the technical parameters used. But you can also dig deeper into these data sets and look at the actual curated cell types that were identified within each of these samples by exploring the cluster level metadata. You can see here down at the bottom right, one dimension reduction analysis comprised of several samples. And we've curated 9 different clusters colored here. I've highlighted cluster 4 and cluster 5 because as part of our curation process, our biologists identified that both of these clusters belong to pancreatic beta cells, curated here and labeled up at the top right. But through the interface, you can also change the expression, the overlay, and look at the clinical parameters, such as disease state, to reveal that one cluster, cluster 4, is populated primarily of pancreatic beta cells from normal control, whereas the other cluster, cluster 5, is populated primarily from type 2 diabetes. You can overlay any of the clinical parameters, such as gender, shown here, or any of the other available. And so this allows you to discover data sets with your cell types of interest, or overlay any of the metadata to reveal patterns and differences between clusters and guide your exploration of these data sets and look for those interesting patterns. To take a step back and introduce you to how QIAGEN OmicSoft curates single-cell data, our data sets are requested by our customers, and our curators inspect each data set request and consider it for inclusion within our database based on a number of parameters. First of all, is the project compatible with our platform? Are all of the key metadata available for integrating these data into the platform and make it interpretable and discoverable within the platform? And are the results actually interpretable between the data submitted to the repository and from the source publication? Or was this just a data dump? After the metadata inspection, our bioinformaticians inspect the actual data and make sure that all of the data that are supposed to be deposited into the repository were actually available. And if those data are available, is the data submission complete? Did they include all of the necessary metadata to parse the data and process it, such as barcodes, UMIs, and so forth? If the data set passes these initial QC steps, our biologists use an extensive curation process to clean up the metadata and centralize and organize all of the metadata, including curating at the project level, the sample level, the technical parameters used for the experiment, as well as any of the clinical parameters available from the data submission into the repository, as well as clinical and sample parameters that might be found within the paper itself. In addition, our curators review the paper and identify the key expected results for these data sets, including what are the cell types that should be discovered within the different analyses, what are the samples that should be compared together in dimension reduction analyses, what are the number of clusters that should be observed in the output from our analysis. And in addition, we note which are the key differentially expressed genes and markers that are key to revealing the different cell types that the authors are expecting from these data sets. At the same time, our bioinformaticians download the raw data and pass these data through a full analysis process from alignment, quantification, post-processing QC, followed by dimension reduction analysis on the relevant data sets and clustering using unsupervised clustering, and then perform statistical comparisons between each identified cluster and all other clusters within the dimension reduction analysis. These data are then used by the biologists for a last round of curation, where for each cluster identified, the curators take the set of expected marker genes and inspect this in an overlay of the clusters and use this information to identify the cell type identity using our controlled vocabularies of each of those clusters. And so this means that you can explore all of our curated data sets and discover the relevant data sets based on precise and manual curation of the cell types that are included. You can filter down on our data sets and explore and discover based on project level metadata, sample level metadata, and the curated cell type metadata. And in the future, we are planning to include additional metadata about how our curators actually approach the curation process and identify these key markers for the different cell types. So you have extensive metadata to help you in interpreting these data sets. In addition, you have access to the full expression data as well as the pre-computed statistical comparisons. So how might you use this in data discovery? Within the platform, let's say that you're interested in liver data sets. You can start with our filters on the left-hand side and filter down on any of the curated metadata. For example, the tissue category, filtering down only for samples that are from the liver. Here we can see multiple data sets from liver from multiple projects identified from the GSE number. And you can see that our statisticians computed multiple dimension reduction analyses, and our curators went through each of these clusters and identified the cell types for each of these clusters, which we can explore here. For example, if you're interested in hepatocytes, you can quickly discover that the hepatocytes were identified in this data set, and this data set. And you can identify it through our interactive plotting, explore the details and the metadata details at the project, sample, and the cell cluster level. And you can change the overlay of this information on any of the metadata that have been curated. As I mentioned before, our statisticians perform comparisons between each cluster and all other clusters within the dimension reduction analysis, so you can explore these comparisons directly. In this example, again, we're looking for hepatocytes, and we're interested in what are the key differentially expressed genes within hepatocytes compared to the other cells that you might find within a liver. So we filtered down on only comparisons having to do with liver, and we found out of the over 100 statistical comparisons generated, we found those where the case group of cells were hepatocytes, and found 12 different statistical comparisons between a cluster that was identified as a hepatocyte and other clusters within the data set. We can quickly go from this comparison distribution view and directly explore those full differential expression results to identify those top biomarkers that are upregulated in hepatocytes. We're looking at 2 of the 12 differential expression results from 2 different studies, and we can see by the interactive plotting that I've selected some of the top biomarkers here for one comparison, finding some candidates for hepatocyte-specific expression, including MAT1A, and we can see those same genes were automatically selected in the volcano plot results for another comparison. So these look like some pretty promising candidates for hepatocyte-specific expression. They're consistent among multiple data sets with hepatocytes. You can inspect this further at the cellular level. For example, looking at that top biomarker, MAT1A, we can directly overlay this on the dimension reduction plots themselves and compare this to the metadata that have been curated. Here we can see from those same multiple dimension reduction analyses, we found our hepatocytes, this cluster, these clusters here, and this cluster. And we can see that our gene, MAT1A, pretty precisely is labeling those exact same cell types. But we can see some interesting differences as well. For example, this cluster down here was also identified as candidate hepatocytes, but doesn't have the expression. So you might want to inspect what are the differences between these hepatocytes and these. And then there's this other type of cell, an EpCAM positive liver cell, that nonetheless has very specific expression of MAT1A. So there's lots of information here that can be dissected and explored further to help really understand these data sets in detail. To take another example, perhaps you're interested in macrophages that might be expressed in a multitude of different tissues, including liver. Using the extensive metadata, you can regroup the violin plot, looking for a gene such as HMOX1, and organize it to help understand and make sense of the data sets that you found. So in this example, we've regrouped on the cluster cell type, so we see the specific manually curated cell type of multiple types of macrophages, including M1 and M2 macrophages, as well as the tissue-resident Kupffer cells that are found in liver. And in addition, we've subgrouped these based on the tissue that they were found in, such as liver, lung, and ileal mucosa. And then to help see the consistency of this expression, we can also subgroup further on the source project here, where we've brought together 4 different data sets into a single plot. You can search for multiple genes and look for the consistency and the similarity and differences of expression on any of the metadata that you're interested in. You can filter for projects, samples, and cell types of interest and group on any of the metadata and look for those similarities. For example, these were the outcome of an IPA analysis of macrophages, and we're checking out some candidate biomarkers for macrophages here. And so we can see that some genes are highly specific for a subtype of macrophages, whereas some others are more broadly expressed across multiple types. You can take this analysis even further and search for dozens of genes and explore, for example, the upregulation and downregulation pattern of signatures for macrophages across all of the different statistical comparisons and look for those patterns of expression of upregulation or downregulation in different data sets across the different projects, across the different tissues, and across the different cell types. So you can identify specific patterns and you can make unexpected discoveries about expression patterns in cell types you may not have even expected. And with that, I'd be happy to take any questions.
Great. Thank you so much, Joe. That was a wonderful presentation. We do have a couple questions here. First one here, are the visualizations you showed available in the OmicSoft single-cell LAN platform?
Yes. Yes, all of those were screenshots from the actual application.
Cool. And is there a limit to the number of different single-cell data sets you could compare?
No, I mean, the more you filter down, the more sprightly the response time will be, but you can explore the full data set or you can pre-filter on any of the individual data sets.
Gotcha. All right, great. And can users of the single-cell LANs framework gain access to the raw or underlying data, either project by project or through a data dump or API?
Yes, absolutely. So one of the key features that we have is the ability to explore the database and use this as the starting point, the hub for discovering data sets. And then you can click on any project you're interested, and with a click, download the full data sets, including the raw and the normalized expression values, the metadata, and the differential expression results into an OmicSoft project. And then you can then export individual data sets for your downstream analysis.
Got it. Wow, that sounds really convenient. Okay, here I've got kind of a two-part question. Do you offer? Let me back up. Let me ask it a different way. Is it possible to add your own data into the single-cell LAN framework?
With our service team at QIAGEN Digital Insights, they can build single-cell data as well as bulk data and any number of different data sets into the LANs framework for exploration.
Okay, and so kind of the follow-up to that was, do you offer any kind of single-cell analysis pipeline if one wanted to compare their data?
Yes, QIAGEN Digital Insights has multiple single-cell pipelines. You can use the QIAGEN Digital Insights platforms and algorithms, or the services team can wrap your custom pipeline into pipeline scripts that can be run directly through OmicSoft.
Got it. Okay, cool. So then we have one last question here, and that is, can you explain what the advantages are of manual curation versus AI or machine learning based approaches?
Yeah. Single cell data are tough. It's huge amounts of data, lots of different metadata sourced from a variety of different data sources, different tissues, different experimental standards and methods of curation. And the source authors may be interested in very different projects. They use different terms. There's no control on the terminology. And mistakes happen when you deposit data. And so really the only way to really explore the data and be able to understand it properly is to have consistent, deep biological expertise applied to the curation process.
Great. All right. Well, I think that's it for the questions here today. I thank everybody for joining us in today's webinar. And again, thank you to our speaker, Dr. Joe Pearson. If you have any questions, please visit our website at digitalinsights.qiagen.com. And with that, we wish you the best for the remainder of your day today. Thank you. We wish you the best for the remainder of your day today. Thank you.